some more terms to look into: endocytosis and exocytosis. Endocytosis and exocytosis are two processes by which particles can be taken in through the plasma membrane. Pretty much you can see in the image A that how the particles seem to concentrate around the plasma membrane, and they have formed a sort of like a pit over here, and this pit starts sinking in and becomes an invagination, and eventually a vesicle is formed. This vesicles that is taken in this form is inside, so the term is endocytosis. Endo meaning in, and exocytosis mean outside. Exocytosis would be the process by which this vesicle then can then throw its molecule to the outside, and it does that by the vesicle first again getting close to the cell membrane. I'm looking at these images and there's something wrong that I noticed over here. Well, no, this is just fine. Okay, I can see what has happened over here. It's showing that it's come over here and it attaches to the one region that's missing over here. It's shown as this membrane. It will attach over here and then open up from this side and eventually start throwing substances outside. So that would be exocytosis, because contents are released in this manner. There are three types of endocytosis. I'm not going to go into a lot of depth, but just briefly, sometimes an endocytosis would be helpful by the substance being binding to the surface receptors and pit being forming, as I showed you before. And another form of endocytosis is called as cell eating, in which the food particles or the nutrients are engulfed by the prey. Amoeba is a simple unicellular organism that eats this way. And sometimes you have a larger vesicle forming around the fluid, and that's called as a bulk phase. Quickly, I'm going to skip this slide. Don't need to worry too much about the details on them. Just know what an endo means and what is exocytosis. The last section is to understand how the water moves around the membranes. One of the ways that water can move is definitely diffusion, but sometimes water can also cross a membrane which is very selective. And this flow of the water is always from the higher concentration of water to the lower concentration of water. And in this particular pattern, what is important to remember is the water is passing not just an open chamber, it is passing through a selective permeable membrane, a membrane that has pores in it, and it is selective to what it allows to pass through. For example, on this right side of the tube, you can see this is called as a hypotonic. Hypo means less, so the pressure is less on this side, and it is hyper on the other side, which means that there is more solute particles on the other side. So if you have more solute would be a substance, more molecules, then it has less water. On the other hand, you have less solute, more water. So now think of it, which way water will flow. Water always flows from higher concentration to lower concentration. So water will flow in this path. When it does that, notice what happens to the elevation or the level of the fluid on this part of the tube. It shows that the fluid level has risen, or it rises because water is moving from lower solute concentration, lower solute, or more water. So they're the two important keywords. You got to pay attention when you're doing a quiz or a question on this kind. High water or lower solute are the same environment. So it goes from lower solute to the higher solute, or higher water to the lower water. Let's look at this picture over here that also talks about the same concept, which is tonicity. Tonicity is basically the concentration of the solute, and it will react, any cell will react differently depending on which tonic level it has been placed to. For example, if this is a 2% sucrose solution, then a 2% solution, if it goes into a distilled water, what will happen? Because if it goes into distilled water, it is called as a hypotonic. In a hypotonic environment, water will get into the solution, into this bag, and it will burst it open. On the other hand, if it goes into a hypertonic, which is this concentration over here, the water, because it's hypertonic, there is more water inside the bag than the outside. So water starts leaving the bag, and the shell will shrink. And in the last scenario, if you take the bag and you leave it in the same concentration, actually this is not, this is 1 liter of 2% sucrose, the same solution, there is no change in the shape of the bag because it has the same concentration level from in and out. Red blood cells that are shown in this image show you that they can burst open if too much water is in the blood. Also, sometimes when you have an infection, it's always better to drink more water, because the more water you drink, it will cause the bacterial cell in a hypotonic environment, and they will burst open. In a hypertonic environment, usually shells will shrivel.